For a long time, researchers called parts of the genome, now known as transposable elements, as junk DNA, disregarded at that moment in time as they had found no use in them. However, they turned out to not just be junk, but rather something more important. So, transposable elements make up almost half of our human genome, with 8% derived from endogenous retroviruses, which include long terminal repeats. These long terminal repeats are often found in open chromatin and potentially act as regulatory elements. Endogenous retroviruses originate from infections where the viral fragments interact with host transcription factors via the long-term repeat sequences and then spread by a copy and paste mechanism throughout the genome. In defense of them, host cells evolved mechanisms that may result in their inactivation. So they were once thought to serve no function. Recently, some have been found to act like regulatory switches. A new method to study these repetitive sequences was used by which a subfamily of transposable elements, MER11A, B, and C, were divided based on evolutionary relationships in primates. The study did so by using a lentiviral massively parallel reporter assay. This is a technique used to test the ability of the MER11 in controlling gene expression. Cells were inserted with the DNA sequences and then measured their gene activity. So what happened? MER11G4 had a strong ability to activate gene expression. It had distinct regulatory motifs for transcription factors to bind to. Further analysis in humans, chimpanzees, and monkeys revealed minor changes in the MER11G2 over time. In humans and chimpanzees, some sequences gain mutations that could increase their regulatory potential. So, why is this important? This study highlights how junk DNA can evolve into regulatory elements that has significant biological roles. Tracing the evolution of these sequences may lead us closer into understanding the role of integrated ancient viral DNA. Transposable elements may have important roles in genome evolution. Their significance may be uncovered through further research. At first, I struggled to understand how transposable elements are integrated from viral origins to be utilized within the host. However, reading the academic article in depth enabled me to understand the rigorous testing and analysis used to arrive at that conclusion. Although many parts of the human genome and their functions remain unknown, by understanding the genome's many segments, even the small ones, will eventually lead to increased knowledge of its functions. In conclusion, I selected this article as it introduced me to the evolution of transposable elements. My Gen 2052 understanding of transposable elements was applied to gain greater insight. I can explore more roles in which transposable elements play other than regulation. By reflecting on my knowledge, I gained more appreciation for the otherwise disregarded junk DNA. In conclusion, this article enhanced my knowledge of transposable elements that I learned from in Gen 2052. I encountered a new method of analysis and technique, the lentiviral massively parallel reporter assay, that emphasizes scientific application as a means to discover the world and its biological history. I am intrigued by the endless possibilities of transposable elements that may be unknown as of the current literature.